Hello, today I will walk through how to run the expression analysis workflow. Before that, I will like to show the data panel where data can be downloaded. Here, the data is downloaded in this RNA-seq folder from NCBI SRA database using the download data workflow. This is human RNA-seq dataset, 2 sample groups and 3 replicates in each group. Let's click on the launch workflow now. I will scroll to expression analysis in RNA-seq. It will open to this overview page, and there are other tabs with more information about inputs, outputs and citations. I'll briefly go through this workflow chart. Once we have the FASTQ files, it will be adapter and quality trimmed, aligned to the desired genome, and BAM files will be generated. We can get QC report using multi-QC from the BAM files. We can generate gene counts using feature counts, FPKM files using string tag, and transcript counts using Salmon. And the final step is differential expression analysis. Let's go to the run workflow now. There are 2 modes here. Full analysis, the input files are the FASTQ files here. The recalculate statistics, the input folder will be the FPKM files and the count files for all samples. I am selecting full analysis now. The first input is the directory where the input files are saved. The second input file is the design file. Either you can create the new file. This should contain 3 columns: run ID, sample ID and sample group, or you can select the existing one. Once you select the existing file, it will load the file. The first column is the run ID, which is the SRR numbers in this case. The sample ID will mention the sample information and the replicate information. The third column is the sample group. Here there are 2 sample groups, wild type and knockdown, KD, HBV positive. Select next step. This is a human RNA-seq dataset, so I am keeping the default HG38. And you can also select genome annotation versions, for example NCBI, Ensemble and GenCode. Click on the next step. In the advanced options tab, you can select different algorithms for trimming. You can select whether you want to run the alternate splicing analysis, and depending on whether it's a stranded or unstranded RNA-seq, you can change the parameters here. This is a stranded RNA-seq data, so I'll change the options and click next step. And finally, you give a unique file workflow name and run workflow. I am not currently running the work because it takes a while to perform the alignment of the reads genome. So I will open the already run workflow. Once you finish it shows the completed sign here, and you get different tabs in this completed run. Go to the all files and output folder. In the output folder, the first file you should see is the hr.txt file. This is the list of up and down regulated genes which are statistically significant. It gives the information about the log2 fold change, p-values for each gene, and whether it's up-regulated or down-regulated. Here the important thing to notice is the file name. In this case, the gene list that you see in the right hand side panel, it is the knockdown up-regulated gene set or down-regulated gene set as compared to the wild type. You can also click on this gene to path.txt. This gives information about the gene ontology terms and pathway terms associated with the differentially expressed genes. And you also get StringDB information, that is another database which will give the information about the functional aspects of the differentially expressed genes. Along with that, you will get FPKM files and count files for each sample, the count matrix and the FPKM matrix. You also can get the HR IDA file. You can use this IDA file, load in R and generate custom plots. I will also like to open this multiqc report.html. This will give the statistics about your BAM file and the FastQC results from the FastQ files. So it gives how many million reads were mapped, how many reads were assigned to the genes, and all the other statistics. The last thing I would like to cover is Open Analysis tab. When you click on this tab, it will load a dashboard which shows some plots which are generated from the results from this workflow. When you click on this, it will take a couple of seconds to load. So I have already opened the dashboard. Here you will see different tabs that will show you different plots. Here I am just showing a sample comparison plot, PCA plot, and you can scroll on any of these dots. You can also download any of these plots that are shown here. You can change how you want to view the plot depending on the design file you get. I think that's it for now. Important thing before I sign out, you should at least have 3 replicates in each sample group to perform the statistical analysis using this RNA-seq workflow. Thank you.